Section we will be discussing about the leucopoiesis and the thrombopoiesis. First, leucopoiesis. So we are having first of all I would like to highlight something, that is the lymphocytes is having a different lineage and the granulocyte plus monocyte is having another lineage. Okay, so first I'll be concentrating on lymphocytes, then I will come to granulocyte and monocyte. So first of all, the basic stem cells, which are pluripotent hematopoietic stem cells, this will be divided into lymphoid stem cells and colony forming unit granulocyte monocyte. Now I will be taking this part, especially concentrating on lymphocytes, then we will come to granulocytes and monocytes. Okay, so this lymphoid stem cells will become lymphoblast and further it will divide to become the lymphocytes. Okay, now coming on to the granulocyte monocyte, it will form myeloblast, then it will further divide into promyelocyte and monoblast. So these will develop into granulocyte and monoblast will be eventually develop into monocytes. So now I will be more concentrating on monoblast, then we will come to promyelocyte. This monoblast will become promonocyte, which eventually becomes monocyte, whereas the promyelocyte becomes neutrophil myelocyte, basophil myelocyte, eosinophil myelocyte. Okay, this will then develop into neutrophil metamyelocyte, and eventually it becomes neutrophil. Similarly, in the same way, the basophil myelocyte becomes basophil metamyelocyte and becomes basophil. Similarly, eosinophil myelocyte becomes eosinophil metamyelocyte and eventually become eosinophils. Okay, so now I will be discussing about the regulation, how this is regulated. So, like erythropoietin, it is also having colony stimulating factor, that means CSF, colony stimulating factor. Like the same way, granulocyte colony stimulating factor, there are two, and granulocyte monocyte colony stimulating factors. This will increase leucopoiesis. Okay, so these two factors will increase leucopoiesis, and by rDNA technology we have developed a drug which is called filgrastim. That means by recombinant DNA technology similar to GCSF, that means granulocyte colony stimulating factor, we have developed filgrastim. Underline the grastim, that means granulocyte stimulating. It stimulates the granulocytes. Similarly, through rDNA technology we have developed sargramostim. Underline the granulocyte monocyte stimulating, that means it stimulates both granulocyte and monocytes. Okay, now the interleukin 5 will play a good role, very important role in the development of eosinophils, and the interleukin 3 and 4 will play a very important role in the development of basophil. Okay, interleukin 5 eosinophils, interleukin 3 and 4 basophils. Now the thrombopoiesis, the last and the most easy one. The pluripotent hematopoietic stem cells will form GM CSF thrombolineage, which will develop into megakaryocyte, eventually forming the platelets. So the thrombopoiesis is also regulated. Similar to the erythropoietin, here they are having thrombopoietin. Okay, this thrombopoietin will increases the thrombopoiesis. By rDNA technology, we have developed a drug named L-thrombopag, which is similar to thrombopoietin, which will increase the thrombopoiesis. Okay, the interleukin 11, it plays a very important role in the development of platelets, that is thrombopoiesis. And by rDNA technology, we have developed a drug called opraleukin. So this is the drug which is developed which is similar to action of interleukin 11. Okay.